All right, so day two. We talked about prophase 1. We're moving on to metaphase 1. So pass me another taco. We covered the P. Now we're on to M, metaphase. Check out how these chromosomes are aligning. So during prophase, they did find each other. They swapped a little bit of information during crossing over. Now, as they're lining up along the vertical in this example, metaphase plate, this metaphase plate is going to be vertical. They're going to separate to the left and right once we get through metaphase 1. And no, not metaphase 1, meiosis 1. But take a look at the way they're arranged. Interesting, they're not the same in each of these little cells. In this first one, we have all blue on one side, all red on the other side. This next one, we have 2 blues, 2 reds on each side. And this other one, we have different combination of blues and reds and even more combinations of blues and reds. So they're not the same. The way that they're lining up is just kind of random. And this is called independent assortment. They are sorting themselves on that metaphase plate, independent of one another. They're not just sitting there thinking, oh, shoot, that one right there is red, so I should probably go on this side. No, they can't think. They're just chromosomes. However they line up along that metaphase plate is totally random. This gives us a second opportunity for genetic diversity. All of these cells are going to have different combinations. Not only did they have that crossing over swap of DNA to create more diversity in traits there, but also the way that they're lining up to be separated in the next step of anaphase is changed also by the way they're lining up along that metaphase plate. So this is our second opportunity for genetic diversity. All right, anaphase. It's a little bit different than mitosis. So up in the top right corner, this is what meiosis, anaphase 1, is looking like. Down at the bottom, this is what mitosis, what we already know during anaphase, looks like. There's a difference. Looking at the way these chromosomes are getting pulled to opposite sides of the cell. In meiosis 1, during anaphase 1, whole double-stranded chromosomes are getting separated. During mitosis, we're popping them open at that centromere and pulling them in half, creating single strands. But during meiosis, anaphase 1, we're just going to separate those chromosomes. So here, for example, here is the metaphase plate. This is where they're going to be separating. We're just going to move them apart. So each half is going to get one big one, each half is going to get one middle-sized one, and each half is going to get one little tiny one. We're not pulling apart those double strands, we're just moving those homologous pairs to different sides. Then we get to telophase. We finally pinch off. It's kind of wrapped up, and also with cytokinesis, we're pinching off, creating 2 separate cells, which happen to be haploid. They are double-stranded, they're still an X, but there's only one of each chromosome. There's one big one and one small one and one big one. They are considered haploid, one of each chromosome. We created genetic diversity 2 times. Once in prophase 1 with crossing over, mingling those tetrads, getting different combinations, and also in independent assortment. During metaphase 1, the way they're lining up along the middle during that metaphase plate, flip-flopping any which way, totally random. Again, it could be this one was 2 reds and this was 2 blues. It just so happened that they lined up in a way that it was one of each color. Totally random, sorting themselves independent of one another. They're not thinking, oh, this guy over here, I don't want to be with them, I'm going to go over here. No. However they line up, totally random. All right, so we finished up meiosis 1, the first stage. We made 2 haploid cells of double-stranded chromosomes. Now we're going to move on to meiosis 2, the second part of meiosis. We did interphase, prophase, metaphase, anaphase, telophase. Wait, we're going straight into prophase. What happened to interphase 2? In the quick right, under column L, why are we skipping interphase 2? Why do you think? Use a little bit of your critical thinking. What's happening during interphase usually? Why wouldn't we need that to happen before prophase 2? We are preparing for division. We're getting bigger. We're duplicating organelles. Then as we move into S phase, we're starting to replicate DNA. Then we're going through checkpoints. Awesome, awesome. Thank you, Declan. No worries. If you want, you can type it in the chat. Cool, cool, cool. So like one of the main purposes of interphase before, we start mitosis or meiosis, obviously it'll spend a lot of time in G0, which is the rest. But once we leave G0, enter G1 phase, what are some of the things we're doing? After G1, we enter S phase, then G2, then we enter the division process. So the main reason, let's see what we have to say. Because our cells already prepared, we didn't need to multiply DNA since we're not making identical copies. We already did interphase. I mean, we're doing twice of everything. Why wouldn't we just do twice of interphase? I love what you had to say there, Elise. Declan was also typing in the chat. Awesome stuff. Once you're done typing, please make sure to hit enter so I can see what you wrote instead of just a weird gray box. What it really comes down to is we don't need more DNA. We already replicated what we need. That's the main, like getting super simple about interphase. But, we already prepared during interphase 1. We're just going to keep going. We're not pausing to take a break. We're just powering through. We don't need to prepare anymore. We're already good to go to move on to meiosis 2. So mostly, we don't need more DNA. We don't need to synthesize or replicate any more DNA. We're already in the middle of this process. We're just going to keep going until we get our end product of haploid cells. Sorry about that. I live on a busy street. All right. Now let's see what's next. Meiosis 2 should look very familiar. We're separating those sister chromatids. It is exactly like mitosis. Hooray, we don't have to learn anything new. Just apply mitosis to meiosis 2. It looks like it's repeating, but that's because those are the 2 cells we created in meiosis 1. So it's just happening concurrently, simultaneously at the same time. They're going through these stages. We're going to have those centrioles move to opposite sides. Then during metaphase, we're going to line up along the plate, down the middle. Then during anaphase, we're going to attach to those centromeres of our chromosomes, pull those sister chromatids apart. Then in telophase, we're going to start pinching off, going through those processes of reforming the nuclear envelope. Those centrioles kind of calm down, their job is done, start to unwind that DNA. Now we have 4 haploid cells. So at the end of meiosis 1, we had 2. Then those 2 split off to go into their own meiosis 2s. And those split up into 2 each, giving us 4. So we started with 2 haploid. Yes, they are still haploid. We had 1 copy of each chromosome. They just happened to be double-stranded. Then at the end of meiosis 2, the end of the whole meiosis process, we split apart those double strands into single strands. They are still haploid. There's still only 1 copy per cell. So let's wrap it up, put it together, sum it up here. We start with 46 chromosomes each, normal human cell. Then those sex cells need to go through meiosis to create our gametes, our sex cells, eggs or sperm. After meiosis, we know that they have half the amount of genetic material. They are haploid. They have 23 chromosomes, only 1 copy of each. Then they will go through fertilization, give us our zygote, our little fertilized egg here. And hey, once that zygote gets fertilized, it goes on its own adventure. It starts going through mitosis, replicating, and we form a little embryo here, which in turn develops into a fetus. Once that fetus is fully grown, we got ourselves a little baby. All right, last part here. Last bit about how we get all of this genetic diversity. Why don't we look like our siblings identically? We do have similarities. So we have, as females, I have, when I was born, about 2 to the 23rd eggs. Females are born with all of the eggs they will have their entire life. And males produce about an equal amount of sperm. They can produce sperm daily. A little bit of a difference there between males and females. But multiply those together, there are over 70 trillion possibilities of these combinations between just an egg and a sperm. We don't know which egg is going to come out from the ovary. We don't know which sperm is going to come out of the testicle. Random egg, random sperm. They already had some mixing of traits, swapping of genetic material during crossing over. All of these things independently assorting during my metaphase 1. Also, we got this random fertilization happening. Random egg, random sperm, meeting up, creating that zygote, fertilizing that egg. Still half and half, but we don't know which genes are which. So for example, here, this guy is going to reproduce with this lady. Their kids, while they look similar, they have some features in common. You can tell the difference. They are different people. Same with this couple over here. This guy is going to reproduce with this lady. Their kids, similar features, but they are different people. You can tell the difference. They're not identical. We had some genetic diversity. Here's a side-by-side. Again, just sort of like a big difference here. Mitosis only goes through these steps once: prophase, metaphase, anaphase, telophase. We start with diploid cells. We end with diploid cells. We have 2 copies of each chromosome during mitosis. Meiosis, on the other hand, start with a diploid. At the end of meiosis 1, we have 2 haploids. At the end of meiosis 2, we have 4 haploids. The only difference between the end of meiosis 1 and the end of meiosis 2 is how many strands that chromosome is. One is double-stranded, one is single-stranded. Here it is in words. Again, repetition, repetition, repetition. Mitosis, there's only 1 division. Meiosis, there's 2. Mitosis, daughter cells are genetically identical. We're making perfect copies every time. Meiosis, the daughter cells are different. Crossing over, independent assortment, they're creating that genetic diversity. Mitosis produces 2 cells that are diploid. We're starting with diploid, we're ending with diploid. Meiosis produces 4 cells, starting with diploid, ending with haploid. Only 1 copy of each of those chromosomes. Mitosis produces all the other cells in our body. Skin cells, hair cells, muscle cells, liver cells, lung cells, all of those. Except for meiosis produces gametes, or those sex cells, eggs or sperm. Lastly, mitosis, there's no crossing over. Meiosis, there is. There's an independent assortment happening there as well. So then, in your quick write, how are we getting this genetic diversity? Why don't we actually look like identical to our sisters and brothers and other siblings? So during prophase 1, how are we getting that genetic diversity? During metaphase 1, how are we getting that genetic diversity? After our gametes, our sex cells, have formed, what's that last way we're getting genetic diversity? Great. Looks like I've got some answers starting to pop in. Awesome, Claudia. Great job. Claire's on it. Awesome. Elise has got it. Loving what I'm seeing. Awesome, awesome. Claudia's on it. Kira's on it. Claire's on it. Andrew's getting it. Perfect, perfect. William's getting it. Lauren's on it. Cole's on it. Yes, yes, yes. Oh, you're making me so happy. I'm so proud of you. The last one's the trickiest one to remember. We didn't spend as much time talking about it. I didn't say it as many times, but we will get it. I will hammer this into your heads. You will definitely remember this forever by the end of this unit. All right, so yes, during prophase 1, we are getting that genetic diversity through crossing over. You got it. During, yes, Claudia, yes. During metaphase 1, we are getting that genetic diversity again, but this time through independent assortment. Yes. And lastly, after we form these gametes, we've done all of our meiosis. What is the last way we get this genetic diversity? Yay, yay, yay. Awesome, awesome. Yes, you got it. It is called random fertilization. Random egg, random sperm meet up, fertilize. We get a zygote with who knows which types of genes and combinations of genes after all of that crossing over and independent assortment. Yes. You guys are awesome. All right. This is all just FYI. I had some questions in 4th period about chromosomes and some chromosomal disorders. This will not be on the test. It's interesting to look at the way different chromosomal disorders come about, what they mean. Generally, they happen through something called non-disjunction. Big word, not that confusing. So during our meiosis, it can happen in 2 different places. It can happen during meiosis 2, where when we're separating during that during anaphase and telophase and stuff like that, we're not getting the correct number of chromosomes in each of those cells. So for example, this is really small. I'm gonna have to make this bigger. Down here on the right, one of them has 3 chromosomes and one of them has 1. They're supposed to have 2 each. It can also happen in meiosis 1 during the first part, where they don't get separated correctly. Then one side just, it's left with nothing from one of the parents. And then it has too many on the other side. Here's a better look at it. Normal meiosis, we're going to get, good equal separation. We're going to end up with, the correct number of chromosomes in our ending haploid cells. But if non-disjunction happens, so this unequal separation of our chromosomes, we end up with things like this one on this side, where we have 3 here and 1 here, when we should have 2 in each one of each chromosome, 2 total chromosomes per thing. Mistakes happen. This can lead to some chromosomal disorders. For example, if this happens on the sex chromosome, an X or a Y, you can get something called Klinefelter syndrome, where a person has 2 X and a Y. They have 3 sex chromosomes. They will display attributes and traits of both males and females. Kind of interesting. Another one that you, I'm sure, have heard of is Down syndrome. This is when the 21st chromosome. So we're looking at our karyotype going across, the one that's number 21. There are 3 chromosomes instead of 2. It should be 2 because we have pairs, but sometimes mistakes happen, non-disjunction, and our kid ends up with too many chromosomes. There are a few other examples. I just wanted to name a couple, one on the sex chromosome, one on the 21st chromosome. If we look at our karyotype up here at the top, the 21st chromosome is loading this guy down here. There might be an extra one that will give you Down syndrome. Again, those chromosomal disorders are not on the test. They're not part of our material. Just had some questions. I find it interesting. Yeah. Any questions, comments, concerns? Do I need to go back over something from today, from yesterday?